higher plants, the sexual reproduction completes in two steps, which is the sporogenesis and gametogenesis. In angiosperm, flowers are the designated structures of sexual reproduction. A flower is termed to be complete if it is possessed of all four parts: the calyx, corolla, androecium, and gynoecium. Androecium and gynoecium refer to the male and female organs of reproduction, respectively, whereas calyx and corolla aids in reproduction. Gametogenesis is defined as the biological process in which the formation of gametes take place. The formation of gametes take place in the reproductive cells of androecium and gynoecium. Now let's talk about the male gamete formation. The formation of male gametes take place inside anther, which consists of two steps: the microsporogenesis and microgametogenesis. When we say microsporogenesis, it is the process of formation of microspores or pollen grains from microspore mother cell inside the microsporoganium or pollen sac of anther is termed as microsporogenesis. The formation of pollen grains take place in anther, which is responsible for formation of male gametes. The young anther consists of homogeneous cells that are bound by a well-defined epidermis. In course of development, the anther becomes four-lobed, and in each lobe, some hypodermal cells become more noticeable than the others because of their larger size and discrete nuclei. The cells of the parietal layer go through a series of periclinal and anticlinal division to form 2 to 5 concentric layers of the anther wall. The primary sporogenous cells, either directly or after a few mitosis, plays a role as microspore mother cells. While microgametogenesis, it is the process of development of male gametes in the microspores, or we call pollen grains, is termed as microgametogenesis. Microspore symbolizes the initiation of the male gametophytic generation. During gametogenesis, the pollen nucleus divides mitothecally to yield the unequal cells. The larger one is vegetative cell or tube cell, which forms the pollen tube, and the smaller one, situated towards the wall, is generative cell, which again divides by mitosis to form two sperms, or we call male gametes. The vegetative nucleus enters the pollen tube. Now, it is known as tube nucleus. The generative tube nucleus enters the tube and divides to form two generative nuclei, which finally form the two male gametes. Anther discloses the following structures. So this is the following structures about anther discloses. First, we will talk about anther wall. It is the fully fledged anther wall. It is consists of an epidermis accompanied by a layer of endothecium. Second, we call epidermis. It is the outermost layer of anther. Third, endothecium. It is normally the endothecium is single-layered and emerge from the parietal cells. Fourth, we call this middle layer. It is 2 to 3 layered and emerges from parietal cells. Fifth, we call this tapetum. This is the innermost layer of anther wall, and arises from parietal cells. It completely surrounds the sporogenous tissue. It is made up of single layer of cells, and each cell contains dense cytoplasm and ovus nucleus. And last, sporogenous tissue. It is the directly play role as microspore or mother cells, or it is called pollen mother cells, or they may go through a few mitosis before entering meiosis. In a meiotic division, each microspore mother cell yields 4 haploid microspores. Microspore tetrads represent the group of 4 microspore. Now, let's talk about the female gamete formation. The formation of female gametes take place inside the ovules, which consist of following steps. First, the megasporogenesis, and second, the megagametogenesis. When we talk about megasporogenesis, it is the process of formation of megaspore within the megasporangium or ovule is called as megasporogenesis. The ovule emerges as a small mass of homogeneous tissue and the placenta and the ovary. Integuments arise near to the base of this tissue, which forms the nucleus in mature ovule. Although the integuments commence later, they grow rapidly than the covers it almost completely, omitting the region of the micropyle. In some others, the hypodermal anchisporal cell divides obliquely, cutting an outer parietal cell and an inner sporogenous cell. The parietal cell may either remain undivided or go through a few periclinal and anticlinal divisions so that the sporogenous cell becomes embedded in the nuclear tissue. While if we talk about megagametogenesis, it is the process of development of female gametes in the ovule, or megaspore is known as the megagametogenesis. The megaspore or marks the initiation of the megagametophyte generation. In the egg apparatus, larger cell is called the egg cell, and the linked comparatively smaller cells are known as synergids. In this way, embryo sac is formed. At the same time, the tissue of nucleus elongates and forms funicle. The entire structure is now termed as an ovule or megasporangium. That would be all about gametogenesis and sporogenesis.